Hello and welcome to VetCov19 channel. Today we have a very interesting guest, Ali Harandi, who is going to talk about vaccines. My name is Jan Lötvar. I'm professor at University of Gothenburg in Sweden. Ali, welcome to the channel, and please introduce yourself and tell us what you're doing.
Thank you, Jan. I am Ali Harandi. I am a docent and head of Vaccine Evaluation Research Lab at Sahlgrenska Academy in University of Gothenburg. We have been doing vaccine research for the past 20 plus years, and I'm happy to be in your program. Looking forward to the discussion.
Thank you, Ali. We met a little bit by accident via the internet, via cyberspace last year when my lab was starting to develop a vaccine candidate, and I reached out to you and tried to learn a little bit more about vaccine development. So you've been involved in vaccine development for many years, and there's been some very rapid vaccine developments happening over the last year from many different companies. But could you just explain how a vaccine, a first vaccine against a disease is developed, the time span and the complexities that includes?
Traditionally speaking, vaccines were developed through a very empirical approach. And if you go back to old days, Louis Pasteur way of thinking for making a vaccine was following the guideline of 3I, meaning isolate, inactivate, and inject. It was a very simple procedure that you see what is the bug responsible for the disease, isolate it, inactivate it by heating it up or later on autoclaving the bug, and then injecting to people. And surprisingly enough, has proven to be a very efficient way of making a vaccine. And nowadays we have a lot of traditionally made vaccines that are based on empirical approaches. Later on, by the emergence of new pathogens and the fact that traditional ways of making vaccines were no longer efficient in providing immunity, people started to think about rational design of the vaccine or engineering vaccines so that we know exactly what we're doing. The other point that people say is that easy ones already taken, difficult ones remained. So which could also be one of the contributing factors why we have several vaccines made in very short period of time, whereas other vaccines are taking much longer time, HIV being one example. Old days used to be that following the empirical approach, making a vaccine was not very difficult. Now, by enhancing the understanding of how immune system operates and new technologies emerging, we are in a better position to have more rational design of vaccines. If we discuss how vaccines are made today, it's a very long procedure. We are now discussing pre-COVID era and what happened pre-COVID, and then I contextualize COVID vaccines with regard to the recent development in terms of technology and what we have learned from HIV research and HIV vaccine. It's a very long process. It usually takes around 10 to 15 years to make a vaccine from scratch. If we go about this novel way, which is reverse vaccinology, we start with the genome of the bug and then see what are the main components of the bug that could lead to pathogenesis of the bug, and then try to elicit immunity that targets those critical elements of the virus or pathogen in question. So the procedure is that we define what components of the bug, in this case virus, is to be targeted, and then making the vaccine using the available platforms. Old days it was inactivating the virus. Later on, we have seen the technology through which we can passage the virus repeatedly so that the virus loses the virulence factors, so it becomes not harmful to the body, but at the same time, it can replicate and it can activate an immune response, which turned out to be very efficient and very protective. One example of a very successful vaccine based on live attenuated is the yellow fever vaccine, which is one of the best vaccines that we have. So we define the component of the bug, which is important for induction of immunity. And then we need to go through the preclinical studies, which is involving vaccinating mice and later on vaccinating some other rodents, because now the regulators are asking safety in more than one species of rodent. So preclinical studies would include showing that the vaccine is immunogenic, meaning that it can activate immune response. Secondly, the vaccine can basically protect in a challenge study. So we need an animal model which is resembling what happened in human, and then we should achieve protective immunity in animal model. And then toxicity is very important. So a lot of attention is given to the safety of the vaccine. And once we have all the documents ready preclinically tested, then they are submitted to the regulators. In Europe is European Medicines Agency, in Sweden is Läkemedelsverket, and in the US is FDA. So they go through the documents coming from the preclinical studies and give a signal whether or not the vaccine candidate can enter human clinical trials. And in the clinical trials, we usually have 3 or 4 phases of the clinical trials. Phase 1 and phase 2, they are merely safety assessment of the vaccine. And for vaccines, safety comes first. And if the vaccine is safe, then the next question is whether or not the vaccine is efficacious or not. Phase 1 is usually a small scale involving less than 100 people. It could be even less than 50 people involved. And then what we produce is basically safety dossier of the vaccine, which would be submitted to the regulators. And if the vaccine proven to be safe in the phase 1, it will enter phase 2 clinical trial with a larger size of the population. And then the primary endpoint again is safety of the vaccine. But during the course of phase 1 and phase 2, of course, we can also generate some immunogenicity data. And if the vaccine trial is taking place during a pandemic, or if there would be an epidemic, you can also generate some preliminary efficacy data of the vaccine. But these are not the primary endpoint of the vaccine trials for phase 1, phase 2. And if phase 2 is a success, then regulators, if they provide green signal, then you can do a phase 3 trial, which is evaluation of the efficacy of the vaccine, in which you are dividing vaccine recipients or participants into 2 arms. It could be more arms, but in general, you have 1 vaccine arm, you have 1 placebo arm. And in placebo arm, you are basically using the diluent or the buffer used for the dilution of the vaccine. And then you have the vaccine arm. Vaccine arm could be subdivided into different arms receiving different doses of the vaccine, or receiving repeated doses of the vaccine versus single dose. And then at the end of the day, you will see how many people are contracting infection in 2 arms, vaccine arm and placebo arm. And then the efficacy, which is being reported, is basically reporting how many people in the vaccine versus placebo arm contracted the infection. And you see the efficacy ranging from 60% to 90% or more. So this is how it works. And once the phase 3 trials are completed, they are submitted to regulators, and they go through all the safety again and the immunogenicity and efficacy data. And depending on the target product profile of that given vaccine, the vaccine will be given approval. So this is the process through which one can achieve approval of the vaccine. And when the vaccine is approved, then it receives pre-qualification, and then it gets marketing approval, and then it can be given to people in different countries. However, regulators would continue assessment of the vaccine even after the approval and after the licensure. namely that they look into the safety, they look into the efficacy of the vaccine, even when the vaccine is licensed and is being used in different countries. And some of the vaccines may get withdrawn during the phase 4. We had, for example, a rotavirus vaccine that was withdrawn during the phase 4 because regulators have gone through the efficacy and safety data and realized that the vaccine is not really giving rise to the expected efficacy. So the vaccine can also fail even after the licensure. So this is a very kind of lengthy and tedious procedure, and there is a lot of failure. So it's more like a funnel. You have a large number of vaccine candidates that are entering preclinical stage, and then several of them fail during the animal testing. And the ones that make it through the animal testing, they enter phase 1. More would fail during phase 1 and then phase 2. Basically, the success rate is very small and it's a very tedious procedure. However, regulators, in particular FDA, puts forth a new way of approval, and it's called emergency use authorization. During the pandemic, some of the vaccine candidates, if they show certain level of efficacy, and of course, if the safety profile is acceptable, they can receive emergency use authorization. Namely, the vaccine can be given only during the pandemic, only during the emergency. When the emergency is over, the vaccine has to go through the full approval. The example of which is COVID vaccines. Before COVID, we had two Ebola vaccines received emergency use authorization. And when emergency is over, you cannot use the vaccine in any countries. So this is the reason, for example, that Merck, which is behind the Ebola vaccine, is now asking for the approval of the vaccine, even though the vaccine has been given in West Africa. So this is the procedure. Now we are reaching the era of COVID, and what happened with COVID, that in 9 months we had several vaccines being authorized, not approved. So we have to make distinction between approval and authorization. So people ask the question, what happened with COVID? I mean, you say that things take a lot of time. So how on earth you can make several vaccines authorized within 9 months? So I think the answer to this question is, number one, we have to remember that technologies that used for developing COVID vaccines, in particular, we have Adenovirus platform and we have mRNA platforms that are used for the authorized vaccines. So there are a lot of research done way before COVID pandemic. And a lot of R&D done for HIV vaccine has been leveraged for developing COVID-19 vaccines. For example, since 20 years ago, we have seen that adenovirus vector has been heavily used for making different vaccine candidates for HIV. And this knowledge has been generated and has been available when COVID hits us. Then mRNA vaccine, over 10 years of research has gone into making mRNA vaccines for different pathogens, Zika and Ebola and influenza. A lot of vaccine candidates were made. But they have not really reached advanced clinical trials due to the reason that those diseases were not really public health concern, global public health concern. And since COVID has this characteristic of being a pandemic, a lot of attention has been given to COVID, and therefore R&D for COVID vaccine has been heavily accelerated, but building upon what has been done in the context of HIV and other vaccines.
Yeah, so that's very clarifying. One of the aspects I was reflecting over is that you discussed only briefly is the hard work that happens before you go into proper testing of a vaccine candidate that you aim to get to the clinic, and that is the research phase. First designing it, and then testing it in mice, and then making possibly multiple different variants of the vaccine before you can go forward and pursue work. And I've realized that in our own research, the collaboration we have ongoing, that the very first version we made was efficient in making some antiviral antibodies, but not sufficient concentration. So we're redesigning the approach, and it's becoming more effective. The vaccine candidate is looking more promising, but it takes actually years. Just that discovery phase also takes years, and then you have the drug development phase that takes multiple years usually, and you have to develop, of course, a very clear process of how you make the vaccine. It's called GMP, good manufacturing practice, and that has to be followed exactly. It's very, very diligent protocols for that. And after that, every batch you make, which can be huge, can be thousands and thousands of vials, then you have to quality control it, QC. And only until then can you distribute the vaccine efficiently to the clinical trial. So, and some of the vaccines that now are available have issues with storage. They need to be stored in very cold refrigerators, and there are many hurdles and many learnings to be had before you can actually really pursue a full vaccine clinical trial. So, any comment on that? I'm sure you have participated in many such studies.
Absolutely. I think it's a very good point that you raise, Jan. And oftentimes the mistake that people working in academia makes is that they start off by making vaccine candidates without respecting requirements of the regulators. Even if vaccines work beautifully in different animal models, if the vaccine preparation is not compliant with GMP guideline, then they have to reproduce all the data that they have generated. So meaning that a lot of research can go into vain, basically, because the preparation was not GMP compliant. So they have to create all the immunogenicity data, all the protection data, everything, even safety. Of course, safety is very important if GMP compliance has not been practiced. So I think this is a very good point that for scientists working in the field of vaccine to make sure that they are basically abiding by the requirements of the regulators in terms of good manufacturing practice and all that. Otherwise, one has to start all over again. And all the data have to be basically regenerated. I think it's a very important point. And scientists oftentimes, they are not familiar with the GMP procedure. And I think the crosstalk between industry and academia is still very weak. So basically, academia is generating a lot of science and industry is making the vaccine, but this crosstalk is by and large lacking. And this could be one of the contributing factors that why some of the seemingly promising candidates would never fly. We generate a lot of publications, many of which would never really get into the arm of people, right? So because oftentimes we don't know how to do it because we are not familiar with requirements of the regulators. And I think your point is very well taken here.
There are a few things that are required to translate academic science to a drug product. One is it needs to be patented, right? If it's not patented and protected with patent, the investors are not so interested because then anybody could copy it and repeat it. So that's actually something that is quite important, and Sweden especially is not very good at helping scientists sort that out. We don't have a system within the universities that help us with that. We have other, but if we ourselves have the capability of doing that, then we have an advantage because we keep the patent ownership as scientists. What was the other thing I was going to say? Yes, and then it's manufacturability, right? Thinking through, of course, we take shortcuts in academic research. we don't have a GMP facility, which means it needs to be a clean room, basically, and have very strict protocols to make that that vaccine candidate or drug candidate for that matter. But what we do is we can outside of a GMP structure, we can test many different alternatives, and then you have to, of course, test them in the GMP environment.
No, absolutely. I think this patent and IP protection, I think, is extremely important, and oftentimes is being neglected by scientists, because I think the metric for scientists and the metric of success is publication, and of course they push for publication, but they are not mutually exclusive, as we know. Even a manuscript as such can be filed as a provisional patent, so that you claim the priority, and then of course you are free to publish from the day that you kind of submit the provisional patent application. So even though there are some guidelines for patent for which perhaps many scientists are not familiar with, and you need, attorneys to help you with, writing a patent and all that, but even in the form of manuscript, you can claim prior art and basically protect your intellectual properties, so that you can keep your candidate alive. Because eventually, whatever we do in university, I think it has to be translated into public health and helping people, right? And if we just go about publication, I think we are not achieving the objective for which we are kind of mission to do. And I think it's a very important point that if there is an idea which is novel and innovative, I think needs to be protected. And I agree with you that there is no mechanism in place to support that. And it's, I think, down to scientists taking initiative in protecting their findings. But I think it's extremely important.
Yeah, and of course, out of the blue or blue sky research, which was without any necessary purpose, sometimes is translated to something. Just a curiosity is teaching us things from the research we're doing, and then you discover things that could be applicable to clinical or translation eventually. But that's not the main topic for today. That's a whole different philosophical discussion, how to translate research to public health use. But let's go back to the vaccines we have available today for COVID-19, and discuss the different types of vaccines and how they kind of work, and how they come about to be available for us in such a short time, Ali.
I think we were kind of lucky that the sequencing was done more or less immediately after the appearance of the outbreak. And I think provision of the sequence of the virus was, in my opinion, a very important step towards making vaccines. So this became available, and of course, the primary target was the spike protein of the virus, because it's important in binding to human ACE2 receptor, and that has become the primary target for the vast majority of the vaccines, if not all. And the ones who have made it to the end, being authorized by regulators, they are all based on a spike protein of the virus. And the virus is in trimeric form. So the trick they did was to stabilize the protein that they make, so that the spike protein, which is given to people either in the form of mRNA vaccine or in the form as part of adenovirus, later on as protein, is basically resembling the structure of the trimeric form of the spike protein of the virus. So attention has been given to the spike protein. So there are some pros and cons by this approach. The pros, of course, is that you are preventing the virus from the attachment to the receptor on the surface of the human cells. But then this component of the virus is very much mutation prone for obvious reason. Viruses mutate in every cycle of the replication. And many of the mutations are completely nonsense to the virus. And it's not giving an upper hand to the virus in terms of binding to the target cell or basically enhancing the transmissibility of the virus. So they are not really staying, but those that are giving an upper hand to the virus, they accumulate. And the constellation of those preferential mutations would lead to the emergence of variants of concern. We have thousands of variants of SARS-CoV-2, but only a handful of them are termed the variants of concern. And those are the ones that are containing a constellation of meaningful mutations that would give an upper hand to the virus in terms of, number one, binding to the receptor, ACE2 receptor, example of which is the British version. And then, or in giving the virus the capacity and propensity to escape from the immune system, like South African one and the Brazilian one. They can escape from the immune system, from the immune response elicited against the original strain of the virus. So vaccines are by and large centered around induction of immunity towards spike protein, and seems to work against the original strain. But what we are hearing now and the data we see so far, there seems to be lower efficacy against the South African and the Brazilian one, which is a cause of concern.
Which basically means that those that have had the early infection, wild type virus, or the B117 British variant maybe, or have had the current vaccines might not be protected fully against those variants of virus, even though we hope that the severity of disease might be smaller because there's still some protection, right?
Absolutely. It's a good point. We have real life data in Manaus in Brazil that 75% of the population in that Amazonian region in Brazil, they contracted infection during the first wave of COVID-19 in 2020. Data are published in Science, 75%. And then when the Brazilian variant of the virus started to dominate, those people got reinfected. So this is, I think, a real life example that immunity to the original Wuhan strain of the virus is not giving rise to full protection against variants of concern. In this case, the Brazilian variant, and the South African one is also having the same issue. Less of a problem with the British one. It seems that prior infection with the Wuhan strain can achieve, very good level of immunity against the British variant. Same problem, I think, concerns the vaccines that are made on the basis of the genome of the Wuhan strain. And the data we have seen is that it could be several-fold reduction in the capacity of the vaccine-induced antibody response in neutralizing the South African and the Brazilian one. So I think the concern is real, not only for those who have already contracted the infection, but also for those who have received the 2 doses of the vaccine. As you pointed out, there are some cross-reactive protective immunity that may lead to those people vaccinated or already contracted infection to be less severely ill with COVID. I think it's a very important point that probably people don't get a severe disease as they would get otherwise.
But the whole goal with vaccination is to achieve what is not possible with infection, in my view, which is herd immunity. Now, if people still get infected, not get that herd immunity component, because you will still be infectious, perhaps not as much as you were if you were not vaccinated, but still you would allow the virus to replicate in you, and you would have the risk of spreading the virus to other people.
Absolutely. I think herd immunity is it's more like back of the envelope calculation when they say 75-80% of the population should be either immune through prior infection or through vaccination. Then you can achieve herd immunity, meaning that you can cut the transmission cycle of the virus in the community or in the society. So I think it's the number is very kind of subjective, I can say, because it depends on which variant is the dominant variant in the population, because the transmission rate of different variants vary, and also the age of the population. There are a lot of confounding factors that should be considered when we are discussing herd immunity. But I think the objective of herd immunity is to reach the stage that the virus has little chance of transmission. And now we are seeing a race between the new variants and development of herd immunity and vaccination rollout. So I think we can say that if the speed of vaccination is much higher, now in Sweden, only 8% of the population over the age of 18 has been fully vaccinated. Only 8% is very low. Over 20%, of course, receive the first dose. So with the speed that we have now, we cannot basically achieve herd immunity anytime soon. We are giving more chance to the virus to make more variants, and we give more chance to the variants of concern to dominate. Now we are having almost 100% in the west coast of the country, British variants, and then we have South African variants circulating in the country. I think 20 out of 21 regions in the country, they have the British variants as a dominant variant. But a few months ago, it was 20% based on the sequencing and the SNP PCR data. Mm-hmm. So if the speed of vaccination is not satisfactory as is now, I think we may not achieve herd immunity anytime soon.
So let's go back to the actual vaccines, the vaccine candidates that are being tested clinically. Some of them are approved, some are not, and discuss a little bit how they work. So one of the vaccines we have had a lot of in the UK and in Sweden is the Astrazeneca Adenovirus vector, which is basically a monkey adenovirus carrying the SARS-CoV-2 gene. Can we discuss the different ways by which these viruses are made? Adenovirus, mRNA. Are there any pure protein viruses with adjuvant out there? How do they work?
So the vaccine that we have now authorized, they are either based on adenovirus platform, like the one from Astrazeneca, which is based on the chimpanzee adenovirus, expressing spike protein, with the difference that here they have not really attempted to stabilize the spike protein, so it's not really stabilized spike protein for AstraZeneca vaccine, as opposed to other vaccines that we have authorized. The other vaccine, which is based on human adenovirus, is not chimp adenovirus, is Johnson & Johnson vaccine, is Ad26. The reason to which adenovirus, chimp adenovirus, or human Ad26 selected is due to the fact that many humans, I mean the vast majority of humans, they do have pre-existing immunity to adenovirus type 5. Adenovirus type 5 is giving rise to symptoms like influenza. So it's very much like common cold symptoms that you get. And many of us, we do have antibody and pre-existing immunity to Ad5. And that was the reason that Ad5 platform has failed. It was a big trial, it's called the STEP trial, against HIV, in which they used adenovirus type 5. And due to this problem with pre-existing immunity to Ad5, they found out that vaccinees contracted more HIV infection in comparison to placebo, because apparently this pre-existing immunity to Ad5 helped the virus to gain access into the target cells. So those companies, Oxford and also Johnson & Johnson, Oxford AstraZeneca, so they went for after the ones for which we do not have any pre-existing immunity. Of course, we haven't seen chimpanzee adenovirus in our life, and even in areas in Africa where people live next to chimps, I mean, because they are in Amazonian area or whatever, in other places where they are in close contact with non-human primates, they don't even have pre-existing immunity. It was a very smart move to kind of circumvent the problem of pre-existing immunity. And for human adenovirus 26, oftentimes people do not have pre-existing immunity to add 26. That was the basis. So they say, okay, we use adenovirus for which we have no pre-existing immunity, and then we basically make it transgenic so that it can present spike protein to the immune system. And since the virus has the propensity of insulting the immune system, because in order to initiate an immune response which is providing protective immunity, we have to insult the immune system. If we are going out with just recombinant protein of the spike protein, we would never achieve immunity, because our immune system is evolutionarily designed to respond to insult that comes from the environment. It could be the bugs or whatever, you name it. But those adenoviruses, they have the propensity of activating immune response because they are seen as a non-self. And when they are activating immune response is activated not only against the component of the adenovirus, but also against the spike protein that is expressed. So this is the basis of the adenoviruses that we have. And then we have a third vaccine, which is being reviewed by European Medicines Agency now. And this is the Russian vaccine, Sputnik. Sputnik vaccine is a dual vaccine based on Ad26, similar to Johnson & Johnson, is the first dose. And the second dose is Ad5. I'm not very much in favor of the second dose because of the pre-existing immunity, but they have this dual platform for the Sputnik vaccine that seems to give rise to some good degree of protective immunity. So this is the package of adenoviruses. And then we have mRNA vaccine, which is based on basically a stretch of mRNA, which is designed to produce trimeric prefusenic form of spike protein inside the body. So basically, mRNA platform is taking advantage of the protein machinery of human body in order to make the protein inside the body. And in this way, you don't need to inject tons of protein, and then it becomes cheaper in comparison, perhaps, to the spike protein used. Even though people are using different platforms for spike proteins, it could be even cheaper in some cases if you inject people with protein. But it's not as simple as taking stretch of mRNA, which is being translated to protein inside the body. So there is a lot of research behind mRNA platform. RNAs in general, single-stranded RNA in general, they can activate innate immune receptors, more specifically toll-like receptors. Toll-like receptor 7 and 8 can be activated among other pattern recognition receptors inside the cell. Cytosolic pattern recognition receptors can be activated by viral RNA. And this is a cause of concern because you give RNA to cells in order to translate it to protein and induce immunity, but in turn, you would get a lot of inflammatory response, which is dampening translation of the protein. So that was the problem. And then also safety problem. You give, tons of mRNA, which is activating innate inflammatory response, and then you may end up getting a cytokine storm that you get, basically. So the trick they made was to eliminate those motif in the single-stranded RNA, which is responsible for activation of innate immunity. What they did was to take away uracil and replace it with a pseudo-uracil, which is less inflammatory. At the same time, they introduced a lot of C and G into the motif. They replaced nucleotides with C and G without changing basically the motif in terms of being translated to different amino acids. But they kept the sequence of the amino acids, but put in a lot of C and G and replaced the U with pseudo U so that you can achieve less inflammatory response and you can achieve much higher protein production. So this was a trick that they have done, and this 10 years of research that has taken place way before COVID hit us. Then RNA is very kind of subjective to degradation because it's not protected by any means. So they had to encapsulate it into this lipid nanoparticle, and this is also many years of research because they tried a number of platforms in order to prevent the degradation of the mRNA, and they came up with the lipid nanoparticle. Lipid nanoparticle, number one, is protecting the mRNA from degradation. And number two is extremely important for giving the mRNA access inside the cells. And third, we learned that it's also serving as an adjuvant because it's a little bit inflammatory. There are some problems. Some people have allergic reaction to polyethylene glycol, which is used for formation of lipid nanoparticles, and they may have allergic reaction to the lipid nanoparticle. Even though not many cases of that reported, but if people have allergic reactions to PEG or other components of lipid nanoparticles, they should not take the vaccine or at least consult with their doctors. So this is basically, in a nutshell, the platforms for which we have authorized vaccines. You mentioned protein platform, yes. Novavax, which is Novavax vaccine, which is built upon basically virus-like particles. So basically they made spike protein in a virus-like particle. The structure of the vaccine is resembling the structure of the virus, but this VLP or virus-like particle does not contain any nucleic acid, any RNA of the virus, so it's completely safe. And then they use it together with adjuvant. In this case, is Matrix M, which is, as a matter of fact, was developed in Sweden, in Uppsala, and they are making the adjuvant in Uppsala. So the protein is made in the States, the virus-like particle, and then the adjuvant is made in Uppsala here, and then they are combined and given as a vaccine. Now it's being reviewed by European Medicines Agency. So we have Sputnik, as I explained, we have Novavax, and then we have CureVac, which is based on mRNA platform. These three vaccines are probably next authorized vaccine in European Union, because now as we speak, EMA is reviewing all the documents from these three vaccine suppliers or vaccine manufacturers. So we would be able to see most likely one more mRNA vaccine coming from CureVac, most likely Novavax, which is a protein-based vaccine, being authorized in Europe. Sputnik, I'm not sure, because Sputnik is based on the same adenovirus platform, and there is a lot of issues associated with the use of adenovirus. Namely, we have thrombocytopenia, thromboembolic thrombocytopenia, and then we have CVS issue, all the blood clotting issues, which is seemingly associated with the use of the adenovirus platform. So I think EMA would be very cautious with issuing authorization for the Sputnik vaccine because it's merely based on the exact same platform.
So if you just take a small step back and think about how these vaccines activate the immune system in our body, basically we inject the vaccine intramuscularly, right? And then the mRNA is entering the muscle, the skeletal muscle, or the adenovirus vector is entering the skeletal muscle. And the skeletal muscle machinery is then protein machinery, ability of making proteins is hijacked, and the cells release the spike protein constructs that the mRNA or the adenovirus DNA has been designed to produce. And those are secreted as free protein, I presume, and are then activating, I guess, immune cells locally, but also in draining lymph nodes. So if you get it in the shoulder, it's probably axillary lymph nodes. Is that a correct way of thinking?
Absolutely. I think this is the way that we get production of protein. And of course, the question is how the immune system would be activated after production of the protein. So I think those platforms, they are relatively inflammatory. Adenovirus and also mRNA because of the lipid nanoparticle, even though they attempted to minimize the inflammatory propensity of those platforms, but they are still inflammatory. And we should keep in mind for a vaccine to work, we need to have some degree of inflammatory response. But the difference here is that the inflammatory response has to be transient. It has to be short-lived. So we cannot afford to have cytokine storm. We cannot afford to have a very long-term inflammatory response, because then the sequelae would be extremely bad, I would say. So what is the target of the vaccination is to achieve inflammatory response, which is relatively localized and relatively short-lived. And this is exactly what is happening. So we have adenovirus, we have mRNA, as you explained. Protein is being being expressed and secreted because they are made to be secreted. And then the inflammatory response initiated at the site of injection would lead to chemoattraction of the immunocompetent cells, antigen-presenting cells coming to the site, picking up the antigen, migrating to the draining lymph node, where they can initiate T cell and B cell response.
Exactly, and that's where we get the antibodies produced. That and in other parts of the body from the B cells and plasma cells. B cells evolved to plasma cells. So if I haven't really looked into the protein vaccine now that might be coming, but they don't need to be injected intramuscularly, I presume, because the muscle is not could they just as well be given in subcutaneously to there meet the immune cells and there be transported by antigen presenting cells to the draining lymph nodes?
It can be given. We have we don't have that many vaccines given subcutaneously, but we have a lot of vaccines based on recombinant protein given through the muscle. So basically, what happens is that you give recombinant protein in conjunction with an adjuvant. And oftentimes you use alum, which through different mechanisms can activate innate immunity. By and large, basically inducing apoptosis and necrosis. When we give alum, it kills myocytes, basically. And by apoptosis and necrosis, you can initiate innate immune response, innate immune cells coming to the site, and then they can pick up the antigen and migrate to the regional lymph node where they can start an immune response. You can give it intramuscularly. You can also give it subcutaneously. The type of adjuvant you use could be different because alum is not designed much for subcu, or definitely not for intradermal immunization. It's usually used in the context of intramuscular injection. But the difference here with the protein-based vaccine is that you don't really need to do any translation to protein because protein is already there. You have adjuvant, which would serve as an activator of innate immunity, and then antigen is there at the same time. Then you can initiate an adaptive immunity.
So another thing that has been discussed is mucosal immunity or mucosal vaccines, and you are one of the world experts in that field. Would you think that an inhaled or nasal spray vaccine could be an alternative to intramuscular injection?
Thank you for the compliment. I don't think I am, but I've worked a little bit. I have some interest in mucosal immunity, and I think it's very interesting and it's very sad. I can tell you why. We wrote an article, no, it was a book chapter a few years ago, and I have a reason why I raised this here. That forced me to go through some old literature and some new literature, and I called it history of failure, because Mucosal immunity is extremely important, as we all know. The vast majority of human pathogens, they take the portal of entry through the mucosa. It could be respiratory tract, urogenital tract, digestive tract, you name it. So think about any pathogen. So the vast majority, they come through the mucosa, right? So having mucosal immunity is extremely important because it's the portal of entry. And when we are discussing prophylactic vaccines, is even more important because you need a very kind of well-equipped army where the pathogen encounters the body, right? For that reason, eliciting mucosal immunity is very important. But there are some challenges to be negotiated when it comes to making a vaccine which gives rise to mucosal immunity, because you want to give the vaccine through the mucosa. So how many vaccines do we have that are given through the mucosa? We have polio vaccines, oral polio vaccine, which is not practiced in Europe, is not practiced in rich countries, it's only developing countries. Used to be practiced here in Europe, but many years back stopped because of adverse events and all that. So we have oral polio vaccines, we have rotavirus vaccines, we have few of them, and then we have cholera vaccine, which was developed, as a matter of fact, in University of Gothenburg here by Jan Holmgren many years ago, 25 years ago. Then we have other vaccines that we have. The other mucosal vaccine we have is the Flumist, the nasal influenza vaccine, which is a live cold adapted influenza. And being cold adapted, the virus or the vaccine has no possibility of replicating in the lower respiratory tract because there the temperature is much higher. So the virus only replicates in the upper respiratory tract and where it can induce immunity. So it seems to be a very good vaccine. It's recommended for people between the age of 18 and 50 or 55, I don't remember. It's not given to young people and to old people, but we have vaccines that are given through the mucosa. But we do not have a single vaccine which is containing any adjuvant given through the mucosa because we do not have any mucosal adjuvant. Why don't we have mucosal adjuvant? Because mucosal surfaces being exposed heavily by tons of other things which are not pathogenic, they are having a state of tolerance. They are having a state of unresponsiveness because the vast majority of things that are coming across the mucosa, they're not really pathogenic. Therefore, the mucosal immune system is having a state of unresponsiveness. If you put in the protein up in the nose or you digest protein, nothing would happen. You swallow spike protein of SARS-CoV-2, I can guarantee you get zero immune response and nothing would happen because the state of unresponsiveness. So you need to use an adjuvant when it comes to the mucosal immunity, and the problem is we don't have a single mucosal adjuvant approved for human use. There has been 25, 30 years of research going into developing mucosal adjuvant, and we also participate in some of those. History of failure because keeping the balance between safety and potency is the key aspect when it comes to the mucosal immunity. And oftentimes when we are too safe because of the state of unresponsiveness, adjuvants have no potency. And if the adjuvants have potency, they are not safe. There are many examples. Toxin-based adjuvants, they have been given a lot of attention. In particular, toxins coming from cholera toxins and heat labile toxins, LT from ETEC, they have been given a lot of attention. And people even, they made the trick in order to mutate in different locations, the cholera toxin and LT, in order to make it less toxic. We have had one example, which I would just name one of them, because GSK company has spent, at the time was Novartis, before that was Chiron. They spent millions into making LTK63, which is a detoxified version of LT coming from ETEC. And preclinical data, beautiful, no toxicity, beautiful immune response. And then when they have taken it into clinical trials, they observed Bell's palsy, which is the partial facial paralysis. in some cases they have seen, because the vaccine was given through the nose, and the nerve ending of the olfactory bulb hanging in the nose, in the nasal cavity. Anything given through the nose has a potential risk of ending up in the olfactory bulb and initiating inflammatory response that could lead to facial paralysis. So there are some problems there. Keeping the balance between potency and safety is the big issue for inducing mucosal immunity.
I think we have one major topic to further discuss, and that is how the vaccine industry and we as human beings should think about vaccinations in the future, and how to adapt our immune responses to the new variants that may not be fully protecting us against, that we may not have full protection against if we're vaccinated with the current vaccines. So what is the strategy of the companies to do this? We have, as you mentioned, B117, maybe not the most concerning, because the current vaccines do have some protective effect. Where we have the South African variant, we have the Brazilian variant, we have something going on in Los Angeles, a Californian variant, and also we have a new India variant that has just been discussed in the last few days in the press. So, and I'm sure we'll get news of new variants probably every month as long as this pandemic goes on. And some of them are, as you said, variants of concern. Some of them may not be. But what is the strategy we as a society, we as scientists, we as or vaccine companies, how should they approach this? And can we be saved from these new viruses, Ali?
No, I think the variants of concern, you named them, and I think one aspect here is that they share some of the mutations. So it doesn't mean that when we say South African is self-standing. So there is an overlap. For example, I think if I remember it correctly, the NY mutation in the spike protein is shared between all the variants. And then what happened, evolutionary thinking, NY has given the propensity to the British variant to bind firmer to ACE2, and that mutation remains across the board for all the variants, but they kind of add to that mutation, for example, having EEK, which is giving immune escape propensity to the South African. And then the Brazilian one has also picked this EEK because thinking like human, it can give an upper hand to escape immunity. So there are some common denominators across the board that can be utilized for making spike protein-based vaccine, which could work across the board for the variants of concern. So we can think this way, that there are some mutations that are common denominator across the board. So if you want to make spike protein-based vaccine, which is pan, we should make sure that those common denominators are well represented, because any new variants of concern that happen later on, they would most likely have those common denominators, and they would have more, which would give them, other properties to evade from immune system, or have higher transmission or whatever. I think the safest strategy is to look into all the variants. If spike protein is the point of attention, and I can get back to this point, if a spike protein is the basis, I think what the companies do, and Moderna is testing, made a new variant or updated vaccine that NIH is testing as we speak. And I think the approach is to make sure that we are covering the common denominator mutations across the board, 5, 6.
Sorry, one moment. Something being delivered. Apologies. Short break. You were saying that the variants are probably very similar. The Brazilian one is very similar to the South African one in a way. And they may occur in any society where there's virus replication, right?
Absolutely. So it could be happening independently, the variants, because they are giving a better property to the, I mean, there are different theories. So some people think that the dominance of the variant in different society could be as a result of people traveling. But this school of thought is not really very popular among virologists and evolutionary virologists, so to say, because the exact same variants can emerge in two different geographical locations independently, because we are discussing evolution. And since every cycle of virus replication involves mutation, only those which are evolutionarily beneficial to the virus would stay. So there is always a chance that we get independently exact same variants emerging in different geographical locations. But I think the point here is that if we are to make vaccines against different variants and future variants, I think if the spike protein is the focus of attention, we need to make sure that the common denominators across the board of the variants of concern are taken into consideration in the updated version of the vaccine, because we are increasing the chance of protective immunity. Maybe the future variants of concern contains more mutations, but most likely it contains the previous mutations, and then we are in a better position of achieving immunity. So the alternative approach is that the variants, they are not only having mutations in the spike protein, they also have quite few mutations in ORF1A, and to some extent ORF1B, but ORF1A is also a target of mutation. And
explain what that is. Explain what that is.
So basically, when we are looking into the genome of the viruses, we have open reading frames, meaning the areas that can be translated into the protein and they become part of the structure of the virus. So spike protein is one ORF, which is called ORF3, because we have ORF1, ORF2, and then ORF3 is a spike protein. So the first component of this open reading frame is also a target of mutation. And in order to have a vaccine against COVID that can take care of all the variants, it's probably more safe to deal with the entirety of the proteome of the virus and not only concentrate on the spike protein. So what people are working on at the moment, and I'm also helping a couple of them, is to basically look into the entire proteome, and through the in silico analysis and having machine learning and deep learning, try to define some putative epitopes that could be the target of the immune system. And those epitopes, of course, you can see if they have any functionalities in terms of the pathogenesis of the virus, and design a vaccine which is a comprehensive constellation of all the components of the virus. So if there would be mutation here and there, you are so comprehensive that it doesn't really matter. Because let's say there would be a lot of mutation in the spike protein, but you have some other components that could contribute to the immunity. And this is the concept of making a pan-coronavirus that can cover basically all variants of concern. Spike protein could be a little bit risky, but if you want to do a spike protein, I think we should go for the approach that I explained. But including other components of the virus, I think would give more chance to achieve this broadly protective immunity, so to say.
Yeah, so I conceptually a whole virus that contain all the components could potentially be a nice vaccine. And some of the components do not mutate as much. The M protein, for example, does not, if I understand correctly, does not mutate as much as the spike protein, which is very dynamic and can even mutate within one individual over time, change its characteristics. The one concern I have whether this would work or not is that wild type infection in Manaus, for example, did not protect against the next variant, even though they I presume had antibodies against the N protein, for example. And you know, I'm raising a small skepticism here, although I'm very, very hopeful we'll get a pan coronavirus vaccine, just as we have polio vaccines that are efficient.
No, I think your point is well taken. There is no guarantee if we are targeting the entirety of the bug, the virus, we achieve full protection against all the variants of concern and future variants. I think we don't know that yet, but I think there is one thing that I would like to discuss here, and this is the immune pressure, the impact of immune pressure on mutation of the virus. We have seen this for HIV, for example. It's a fact that by day 20, 25, you have CD8 response, and it takes a week for the virus to make escape mutants so that CD8 T cells cannot target. And then the same story is for the neutralizing antibody, that there would be escape mutant. So if we are targeting the entirety of the virus, then the immune pressure is not on only one component of the virus, spike protein. So the virus, I mean, it's a very oversimplification of the fact, but I like oversimplifying because it helps understanding. So basically, we are distracting the virus. If we are targeting immunity, if we are achieving immunity, if we are inducing immune response towards different components of the body, so the immune pressure would be on different angles to the virus. And the virus, of course, should work economically. It cannot invest on undergoing mutation across the board, because the virus has to be thermodynamically alive, right? So in that sense, I think it's not in the best benefit of the virus to, address all the immune response elicited towards different components. So there would be a distraction and probably spread of the mutation across the board, which would hopefully give a better chance for achieving a broadly immunity. I think, in my opinion, it's better to speak about other components. I mean, look at the vaccines that we have. Live attenuated vaccines are the best. And then next to that, we have inactivated viruses. They work beautifully. They work very well because we are involving, different components, and then these escape mutants have less chance. They get a lot of distraction because immune system is attacking them from different angles. And then they cannot thermodynamically speaking probably afford, or economically speaking, afford to undergo mutations that cover, all the immune responses that are out there. This is what I think.
So that would be the best future vaccine, one that has probably one of the new spike protein variants, and is it an attenuated or killed virus, basically, without RNA inside. Is that
perhaps, or else would be to go for a polypeptide vaccine, which is covering the important component of other proteins which are immunodominant and play some role in protective immunity. It could also be a cocktail of mRNA vaccine, which is covering spike protein, M protein, part of the ORF1. We can use cocktail. I don't see why not. I mean, you can do the exact same thing as you do with the protein with mRNA vaccine to use a cocktail. And I know that, for example, in China and in other parts of the world, they are doing clinical trial based on live attenuated SARS-CoV-2, doing clinical trial based on, inactivated. We have a vaccine. We have inactivated vaccine in China, right? Sinopharm vaccine is inactivated vaccine, even though the efficacy is not that high. But there are tricks. You can combine it with a good adjuvant. You can have, one thing we haven't touched upon is to use a heterologous approach, so that in priming you are using one platform and in boosting you use a different platform. Sometimes you are forced to do that. For example, here, in this country, in Sweden, 200,000 people received the first dose of Astrazeneca vaccine. Below the age of 65, 200,000 people received. And Folkhälsomyndigheten, and the public health agency of the country, a month ago decided to just give the vaccine, the Astrazeneca vaccine, to people 65 years and older. 200,000 people already received the first dose. So what we do with them? So they decided to give them Pfizer or Moderna. Here they say we give 2 doses. In Germany, they say we give one dose. I think Germany approach is correct, because now we are dealing with the shortage of supply, and it's not wise to give 2 doses to people and vaccinate less people. I mean, this is a different, I think, discussion that we can have. But I think now we are forced to do this prime boost approach, heterologous prime boost, because 200,000 people here and many elsewhere, they already received Astrazeneca adenovirus. Now they receive mRNA. We have a chance to see how this heterologous prime boost would perform. And I can tell you that it most likely performs better than using same technology across. But we will see more data coming along very soon, I guess, from forced trials. But Oxford is doing a well-designed trial in which they are combining Oxford vaccine with Pfizer vaccine.
Yes, I've heard about those studies ongoing for several months already. United Kingdom doing different approaches, and they've expanded those studies and get information in a month or so. So right now, what's happening right now, Ali? So for Moderna or for Pfizer BioNTech, it takes a few days to make a new mRNA construct to target another variant. And I presume that those are already being tested in clinical trials. How, firstly, what do you know about that? And secondly, how extensively will those be tested, and when will they be available for clinical application?
Moderna, the Moderna vaccine, the updated version, is being tested by NIH now. And the expectation is that by June, we have the data from this updated version. And I think, like you mentioned, I think updating mRNA vaccine is not taking so much time, so it can be easily updated. The problem, of course, is the shortage of supply for the ingredients of the mRNA vaccine. They could not keep the promise that they made, and this contributed to the shortage of supply in many countries, and especially in Europe, because those vaccines, mRNA vaccines, they are made in the United States, and they stopped the export of the vaccine. And so there are some vaccine nationalism issue that one can discuss in some other occasions. But now there is a shortage of supply. They can be easily updated. They can be easily given to people. And one thing that we have to discuss here, I think, is that data, Pfizer data just published that 6 months into the vaccination, you see 1 log reduction in the neutralizing antibody titer. So still above the level of protection, in vitro level of protection, of course. But so there is a tendency that the antibody response is going down. And Pfizer CEO just a few days ago announced that mRNA vaccines should most likely be given every 6 months or every year, and then they would have the chance to update the vaccine. So we are turning, I think, this whole story into, seasonal vaccines like influenza vaccines, that we need to get an update shot. Number one, because the duration of immunity is not probably long lasting, what we are hearing and what we are seeing. And number two, there are variants of concern. So this would be exactly the same story as influenza vaccine.
And ideally, having a vaccine against the current variant, and then a follow-up with a vaccine that produces the new variant would be an ideal approach. There's a lot of vaccine strategic thinking going on right now, and I think we'll get a lot of new data around that over the next few months.
Correct.
Ali, thank you very much. You have anything additional to discuss here? This was really very, very educational and helpful.
Thank you for having me. I don't have any other comments, and there are some other aspects that require, I think, separate discussion. But thank you for having me, and I think it was a good discussion that we had.
Thank you very much, Ali, for joining the channel and for discussing general vaccine manufacturing and vaccine production, and specifically for COVID-19. Thanks very much.
Thank you for having me.